Good afternoon, ladies and gentlemen. I would like to welcome you now to this continuation of the session here in the Cluster 1 Work Program 2024. I am Bernd Reiner. I'm a policy officer in the Health, Innovations and Ecosystems Unit, and I'm in charge of Destination 5. Destination 5 in the health cluster is the destination which is the technology-oriented one, and it is entitled Unlocking the Full Potential of New Tools, Technologies and Digital Solutions for a Healthy Society. And as such, Destination 5, or all the activities launched under Destination 5, they follow a rather disease-neutral concept, but a technology-specific one. And I would like to enter immediately into the medias res, into the core of the story, and would like to show you or to remind you of the topics which are now open under 2024 in Destination 5. There are actually two topics, a two-stage topic and a single-stage topic. The two-stage topic is entitled Innovative Non-Animal Human-Based Tools and Strategies for Biomedical Research, and it has a deadline for the first stage on the 19th of September 2023, and for the stage 2 on the 11th of April 2024. The instrument is a research and innovation action, and the total budget available is 25 million euros. The second topic, the single-stage topic, is entitled Bioprinting of Living Cells for Regenerative Medicine. It has a deadline of the 11th of April 2024 and is equally a research and innovation action and has also 25 million euro total budget. Now, to say maybe upfront what the overall goal of these topics is, or of the projects which we would like to fund that are submitted under these two topics, the ultimate overarching goal is to provide better and more cost-efficient healthcare solutions which are tailored to the needs of the individual. The first topic, the non-animal human-based tools, is something which should address new tools and concepts, general concepts for biomedical research, which mimic better the human situation. Whereas the second, the bioprinting, is a very technological topic where it really comes to cover the whole suite of the bioprinting process and to give to this field of technology a boost in Europe. So, and now I would invite you to post your questions via Slido and to use this time really to all types of questions you may have regarding these two topics. I would also like to mention that my deputy head of unit, Ion van Hengel, will also be in this call, in this video, and will help out in case there will be ultra-tricky questions. I see now the questions arrive. I see the shoot for the for the two-stage topic, the focus of the project be the reduction of the number of animals used in any area of research, this is actually mentioned in the topic text what the priority should be. It is both the number of animals, but also the severity of the animal suffering, which should be the main criteria for selecting the disease area which you would like to address with these new developed non-animal tools. Another question on the same topic, could tools that address rare diseases be a focus area within a project? In principle, yes, it is not excluded, but again, also what I already said for the first question, the main criterion is the number of animals plus the animal suffering, and also in the project, in the topic text, it is mentioned that the studies should focus on or should help to improve the understanding of disorders which have a high impact on public health. So these together, these two criteria looked at together give already an indication that rare diseases might not be in the first priority line, but a priori there is no exclusion. It will be up very much to the evaluators to decide which concept is a really sound one to respond the best to the topic text.
And if I can add to that, I think it's on the number and the severity of the suffering, of course, that is taken into account. In that sense, it's also maybe important to mention that information on these aspects can be found in the ALURIS Statistical EU database. So if you want to get more information on these aspects, that's the ALURIS Statistical EU database that can provide information about that.
Thanks, Arjen. Yes, absolutely, yes. And if I'm not mistaken, I think the last update was from 2020, and it looks like that very much three disease areas have a tendency to be more or less always on top of the list, which are, I think, the nervous system, cancer, and the immune system are areas which are very, very much prone here in the priority of numbers and also of severity of animal suffering. Okay, now I see a question regarding the involvement of JRC at the proposal stage. Actually, it is foreseen that the JRC is not involved as a partner at the proposal stage, but the JRC is ready to collaborate with any successful proposal, with any successful project entering at the grant preparation phase. So this is the way how this process is normally made up. At the more or less at the end, when there is a short list already of proposals there, the JRC gets in contact with the proposals which are on the short list, and then be it in a board, or be it in a scientific advisory committee, or be it really in a task to be involved, this is then only decided at this stage of grant preparation. Now I see a question on the second topic, on the bioprinted tissue for regenerative medicine. The Availability or accessibility of larger scale bioprinted tissues is mentioned. Does this imply that tissue of larger surface or mass will have to be printed? This refers actually to one of the expected outcomes which are defined for that topic. And it is thought actually that with larger scale bioprinted tissues, it's meant that both larger quantities of bioprinted tissues should be available as an output of the projects, but also larger tissues in surface and in mass, which will in the end allow to construct really complex tissues and going up even to the level of organs. So it's both the number of tissue, the scale plus the mass, which should allow then to really give a boost and to come up in the tissue differentiation to a very high level. The question on the scope for the combination of different bioprinting technologies to obtain fully functional constructs. Does this combination activity have to be included? This refers to the bullet points under scope of the topic, and it is, I think, the last bullet point in this section, where the activities mentioned. It is mentioned in the topic text that most of those activities should be addressed. So it is not an absolute must that this last point is addressed. However, of course, the majority of those activities, they should be addressed in order that we get really a tangible output of the projects which are submitted under that topic. Another question going to the first topic, the non-animal tools and strategies. Should the developed tools and strategies be compared to current animal models in the project? This comparative studies and validation studies are not excluded, of course. They can be part of the project work, of the proposed work. Nevertheless, what should be said that in order to have really a meaningful comparison or validation is that the proposed tools and strategies should be always compared to the gold standard for the particular chosen disease area or approach. This should be the rule. Then I see a question on the animals which are considered to be as animals within the scope of the topic. The Drosophila zebrafish and zebrafish embryos considered as animals in relation to the scope of the topic. To make it short, zebrafish, yes. Drosophila, no, is not considered as an animal. And zebrafish embryos, to my knowledge, are considered animals as in the late stage of their natural development. Then I see another question on the topic on the bioprinting. Should the outcome of the project be free available bioprinted tissue? This should be considered as in a generic sense, actually. Of course, the output cannot be freely available bioprinted tissue, but this refers, if I'm not mistaken, to the first expected outcome of the topic. It is expected that on the long term, with the work of the projects, there is in general more of this bioprinting equipment and bioprinting facilities are available and accessible for biomedical scientists in general. This is the overarching goal of this first expected outcome. Then I see a question on the non-animal tools again. Is it possible to study a field where animal models are not currently used because not relevant? In brackets, personalized medicine. This will somehow be difficult because the goal of the topic is to develop tools and strategies where currently there is a high number of animal models used and there is high animal suffering. So I think that this is rather should be probably the scope of another topic. Here it is clear that the target is to develop biomedical concepts which enable to reduce the number of animals used in the research and also reduce, of course, their suffering. Then I have another question. Will it be mandatory to propose a computational model? What if the envisioned tool will be an organoid? There I can clearly say organoids are very welcome. They are even mentioned under the examples in the text. And it is not mandatory to use a computational model, but in silico models can equally be part of the whole arsenal of tools and strategies which are proposed. Then I see a question on, in bioprinting, preclinical and clinical studies are eligible to be included? Answer, clear yes, absolutely. And then I see, are both calls directed to any disease or tissue respectively? No, as I said at the beginning, this is a particularity also of the destination. There is no clear indication of a disease area or any tissue or organ. This is to be chosen by the applicants and well justified in their proposal. And this is actually then the task of the evaluators to get here priority for those proposals which respond to the topics best in their entirety. Then I see another question on the two-stage, but I think I have replied to that already. And I think we are coming close to an end. So I think I can say that I would thank you very much for the questions, very pertinent and to the point. And I'm very confident that we will get quite some very good and interesting proposals. And I wish you all the best for the preparation and the evaluation thereafter. Thank you very much.